Hello everyone, welcome to MMBS111 Introductory Microbiology. This is mini lecture 13.2, and in this section we're going to be discussing the first stage, the development of lymphocyte diversity, and we're going to be talking about how T cells and B cells differentiate. There are 4 learning outcomes for this mini lecture. First, summarize the maturation process of both B cells and T cells. Second, diagram how lymphocytes are capable of responding to nearly any epitope imaginable. 3, describe antibody variability. And 4, outline the process of clonal selection and expansion. So this image actually comes from your text from section 13.2. It can be found on page 355. And this shows the surface of a T cell. Remember, T cells are going to mature in the thymus. All T cells possess what is called antigen-specific receptor. T cells also possess what's called a CD3 receptor, and these receptors surround the antigen-specific receptor and assist in the antigen binding to that receptor. Helper cells also have what are called CD4 receptors, and this is an accessory receptor protein that binds to the MHC2 molecules. So remember, B cells, macrophages, and dendritic cells are going to have those class 2 MHC molecules on their surface so that they can present antigen to T cells. So the helper cells are going to possess CD4 receptors that are going to recognize antigen that is presented to them by those MHC2 receptors on those cells. Cytotoxic T cells have an additional receptor called the CD8 receptor, and these are going to bind to the class 1 MHC molecules, and they're going to, these are the cells that are capable of killing other infected cells. Okay, so this is the surface of our B cell. Remember, B cells are going to mature in specific locations within the bone marrow. The naive or sort of unchallenged lymphocytes are going to circulate in the blood, and they're going to collect in specific sites within the lymphatic system, specifically in the lymph nodes, the spleen, and GALT that we talked about earlier. And then from those sites within the lymphatic system, B cells will then come into contact with antigens. And each of those B cells will possess a different antigen binding site that's going to recognize a very specific antigen. All right, so this table is also from your text, and it compares B cells with T cells and distinguishes these two types of lymphocytes from one another. So you can see each of these cells mature in different locations: B cells in the bone marrow, T cells in the thymus. They also possess different surface markers. So B cells possess immunoglobulin, that's the antigen receptor. T cells possess the T cell receptor, and then other T cells also possess different CD molecules either CD4 or CD8 or CD3. B cells are going to circulate through the blood in low numbers. T cells are found in the blood at much higher numbers. The antigen receptor on the B cell is the immunoglobulin or the Ig molecule. T cells recognize antigen using the T cell receptor. B cells are found in the cortex of lymphatic organs. T cells are going to be found in paracortical sites, so they are going to be further in from the follicles. B cells do not require antigen presented to them with an MHC molecule, while T cells do. Remember, our macrophages, our dendritic cells, and our B cells are going to present antigen using those class 2 MHC molecules. They're going to present antigen to T cells. The product from stimulation by antigens for each of these is different. So remember, B cells, when stimulated by an antigen, they're going to reproduce, and in that clonal production, they're going to form plasma cells and memory cells. T cells are going to form a number of different types of activated T cells, like our helper cells and our cytotoxic cells, as well as memory cells. Each of these lymphocytes also have different functions. B cells are primarily responsible for production of antibodies that are going to inactivate, neutralize, or target antigens so that they can be removed from the body. T cells, their job is to activate and help other immune cells. They can also suppress or kill abnormal cells. They're also going to mediate hypersensitivity. So we have a number of microorganisms as well as antigens that can induce hypersensitivity. So T cells are going to help to mediate that, and then they are also responsible for synthesizing certain cytokines that initiate different parts of the inflammatory response. Okay, so by the time our B cells and T cells reach their specific lymphoid tissues, each one of them is already equipped to respond to a single specific antigen. And that occurs because the genes that code for the antigen receptor site is very variable. There are 500 different components, different segments involved in this gene, and those segments can be rearranged in a number of different ways. So every possible recombination of those 500 gene segments occurs in the formation of B cells and T cells. So that is going to lead to a huge variety of lymphocytes within the body. It's estimated that humans produce about 10 trillion different antibodies that recognize 10 trillion different antigens. So although these cells have not seen antigen yet, the variability in the segments that code for the antigen receptor is going to allow for these cells to recognize an innumerable number of antigens that can possibly invade our tissues. So this is the B cell receptor. This is the immunoglobulin, or Ig. It is a large glycoprotein molecule. It's found on the surface of B cells. And when it is secreted, it is called an antibody. The immunoglobulin structure is composed of two main sections. We have the constant region, and that region is connected to the cell membrane when it's attached. And then we have this variable region, and that variable region is the region where the antigen will bind. This variable region is the region that because of the number of different gene segments and the way that they can be rearranged is going to result in a very specific shape that's going to recognize a very specific antigen. The T cell receptors are similar to B cell receptors. They are also formed by genetic recombination in this very same way. They have a variable and a constant region. They are also inserted in the membrane of these cells, and they possess an antigen binding site. The receptors on T cells are relatively small. The big difference here is that they are never secreted. So T cell receptors, while they also vary, and they are going to be specific to a very specific antigen, and their variability allows for a huge variety of T cells that will recognize innumerable antigens. These receptors, these compounds on the T cells are never going to be secreted into body fluids. So you're not going to find these in the blood or in the lymph fluid. Okay. So this diagram is also from your text. You can find it on page 357. And this illustrates the process of clonal selection and expansion of both B and T cells. So the process is identical. This is essentially how scientists hypothesize that B cells and T cells are produced with the variability that they are. You can see in step 1 that we have our lymphocyte cell, and it's going to differentiate in either the bone marrow or the thymus, depending on if it's becoming a B cell or a T cell. And as it differentiates, you can see that the variability in that antigen receptor section of the receptor for that cell is going to result in trillions of lymphocytes that are all different. Each one is genetically distinct, that will only recognize a single specific antigen. So you can see the receptors on the surface of these cells are all unique. The issue that presents itself in this hypothesis is the possibility that a B cell or a T cell, because of the variability of the gene segments in the variable region of that receptor, could result in a B or T cell that recognizes self. And we don't want that. So B and T cells that recognize self are eliminated from the pool of cells. So any of these cells that are produced because of the variability that recognize self-receptors, they're going to be removed. And we call this immune tolerance. So we want our immune cells to have tolerance for the receptors on our self cells. So any cells that are produced that recognize self as antigen, so those receptors bind to our self receptors, we're going to remove those cells from the pool so that we're only left with B cells or T cells that have unique receptors that recognize a single specific antigen. When an epitope enters the body and is comes in contact with an immune cell, a B cell or a T cell, that epitope is going to be recognized by the cell with that receptor that recognizes that antigen. And once that recognition has taken place, then that cell is going to produce clones of itself that will recognize that same antigen. So then we're going to have hundreds of cells produced now that are all clones of that first stimulated cell that all recognize this antigen that has been presented within the body. And that is the end of section 13.2. So here's some checking in questions. Make sure you go through these and ensure that you can answer them appropriately. Remember to go back to the beginning of the presentation and work through those learning outcomes. Make sure that you have mastered that information. Here's our critical thinking question. The first human Zika infection was identified in 1957. The first serious Zika outbreak occurred in 2007 on the island of Yap. Since Zika is a relatively new infectious disease, will your body possess a T or B cell capable of recognizing this antigen? And explain your answer. So think about what we've discussed as far as variability and the presence of these receptors on B and T cells and how they recognize antigen. Form your response, and then you can find the answer on the next slide. And hopefully this video was helpful.